Oceanography students. I want to talk a little bit about the oceanography as a science module. So you guys are kind of starting to finish this module. Hopefully you've been enjoying it. It's not super oceanography related, but it is something that you are going to use this information in many, many, many science classes because it's just how to be a good scientist. So here's your steps of the scientific method. The big one that I see students struggle with, and I'm already seeing students, starting their tests and taking their tests, and so, I'm starting to see, some of these same issues over and over, is sometimes students are saying that the hypothesis should be first, okay? The first thing you need to do is identify that problem, because if you haven't identified that problem, you don't have anything to make a hypothesis about. So even though it seems like the hypothesis is kind of something you do in the beginning, and a lot of times you'll write it in your introduction, or it's one of the first things you do when you're like doing a lab assignment, you really need to identify that problem first. In a lab assignment, we've kind of already identified that problem for you. Like, we're going to be doing this. We're going to be testing the buoyancy of those little tinfoil boats that you guys made or whatever. So the problem has already kind of been identified for you. The next thing you're going to do is you're going to gather information, and this information gathering is going to be important for your introduction, which would be nice if I could spell that. So in your introduction, you may have noticed when you did your Stinker Float Lab, you may have lost some points because you didn't cite any sources. So an important part of the scientific method is gathering data before you even start your experiment. Kind of what have others done? What is known about this topic? So you can be informed when you're working on it, because you don't want to test something that 100 other people have already tested if you're designing your own experiment, because, whatever, it's already been done. It's not really interesting or useful. So that means you're going to want to find at least one external source that has information about what you're discussing, and so then you'll want to have those like in-text citations. So, for example, if it was a paper I wrote, it would be like Crodal. Let's say I wrote it this year. It'd be Crodal 2019, and then at the end of my paper, I would have like sources or a citation section, and then I would write my whatever paper it was that I wrote that you cited. So that's kind of an important part of the scientific method. So that's why it's important that you have those sources and you're including that information in your introduction. Then you're going to make that hypothesis. And in general, your hypothesis is usually going to follow that format. Like, if I do this, then this will happen. And so, and you can't really make like a really educated guess hypothesis if you don't do that background research. Then you're going to test the hypothesis, and I think this is the fun part, right? That's your experiment. This is where you're collecting the data. The next thing you'll do is you'll look at those results and you'll analyze them. A lot of times scientists will use statistics. Almost always they'll use statistics. We won't use a ton in here. We might not even use any. But statistics are important. I don't know why I'm having so much trouble spelling today. Then you'll draw some conclusions for that data. Was your hypothesis supported or not? What's going on here, what might be some future research you might want to do, that kind of thing, and then you communicate your results, and that's basically your lab report. So like professional scientists, people that like being a scientist is their job, they actually basically write lab reports. They don't call them that, they call them journal articles, but they're basically a lab report. They have all the same parts of a lab report, and they write those and they'll submit them to a journal, and then the journal will basically send their paper for other people to read. And they'll kind of critique it and that sort of thing. And that's called peer review. And peer review is really important because, you know, you can't just, in order for a paper to get published or research to get published in a journal that's reputable, there's a peer review process. And so that means other scientists are looking at that data and asking questions and having them edit their lab report to make that a little more clear, their journal article to make it a little more clear. And so there's that kind of policing of the scientific community between each other and making sure that they're submitting quality work and their research was done, you know, in a quality way. And so any conclusions that are drawn from that data are valid because, you know, if you kind of made a mess of your experiment or it wasn't really done very well, you can't really draw that many conclusions from that data because, you know, you didn't do your experiment well. So that's kind of the steps of the scientific method. So this should say basics. I was, I guess, getting a little excited for real oceanography. Okay, so quantitative data is going to be numerical. It's going to be numbers. It's when you're collecting data in a data table. You know, you're measuring something. It's what you traditionally think about as data. But there's also a place for qualitative data, and that's it doesn't have any numbers in it, and it's basically, you know, you're talking about like the size, the shape, the smell, the sound. For example, when I did my master's project, I would write, you know, it was outside, and so I would write, is it sunny? You know, is it raining? What's going on? You know, some qualitative data, what the corals look like. And so there is a place for qualitative data. Unfortunately, it's not as easy to analyze statistically as quantitative data. That's why we like to have some sort of numerical data, because it is a little easier to analyze. And I'll tell you a secret, I did not learn the difference between quantitative and qualitative till I was in college. I won an award for a quantitative award, and then all the kids that did, like, mine was a science project, and a bunch of kids that were like, did English projects and stuff won a qualitative award. So it was around that time when I won that award that I realized that quantitative must have to do with numbers and qualitative must have to do with writing. It's a little embarrassing. Now, here's a way that you can remember it. Quantitative has that in it, so you can remember in for number, and then I remember L, or even better, let me erase it a little bit. Go back to my little pen. Qualitative has like lit in it, which is like literature, so it's like writing. So that's kind of how I remember those two now. So hopefully you learned that a little bit before I did. I don't know how I got that far and didn't know that. Now, experimental design. Let's talk about our experimental design. I know I've done a video about experimental design before, but I did want to talk a little bit about your independent, your dependent variables, and your control. So the big thing is that your independent variable is the thing that you are changing, and you can only have one thing. So you can only change one thing, okay? So this picture is an experiment where they have 3 plants, and it looks like they're watering one plant with soda, one plant with juice, and one plant with water. Okay, so what they're watering the plant with is their independent variable. Okay, so the thing that they're watering with, or the substance they're using, or the liquid. Okay. So that's what they've got there. And in here they're saying these are the 3 things. So if you wanted to have a control variable, or your control sample, you might have one right here where this plant doesn't get watered anything at all, okay? And that way you could compare. Okay, so that would be like your control variable. Okay, or your control experimental group, your control group. Let me see if I can fit group down here. Yeah, there we go. Okay, so these three would be your experimental groups, and then that one would be your control. And do you see how we have at least 3 experimental groups? You really want to do that. You don't really want to have like an either or kind of situation. You don't want to have like your control and then water, or your control and then juice. Because obviously watering a plant at all, hopefully, with water is going to be better than no water. So you want to have like different types of liquids, which is what they have here. That way we're comparing these different types of liquids to each other. And then the control is kind of just an example of what would happen if they didn't get any water. So you really want to have more than one experimental group, or more than 2. Now your dependent variable is the thing that happens because of the independent variables. So what are we measuring? We're measuring the height of the plant. You could measure the height, you could measure the health. So if we measure the height of the plant, right, we would be using a ruler or something like that, and we would be collecting numbers. So that would be a quantitative data measurement. Okay, if we were measuring the health of the plant, the health of our plant might be, if it has green leaves, or the leaves turn yellow or brown, or does it start wilting or whatever. But that's going to be more of like, words that we're going to write about those plants. So that's going to be a qualitative measurement. Okay. And then the last thing is you want your experiment, they call it a controlled experiment. And it's not a controlled experiment because you have a control group. It's called a controlled experiment because you want to keep everything as much the same as possible. So for example, if these were your 4 groups, your 3 experimental and your 1 control, you might want to put them in the exact same windowsill. That way they're getting the same amount of sun as much as possible. You want to put them in the same size pot with the same amount of soil. You want the same type of plant. Okay, like you don't want this plant to be a pea plant and this plant to be like a daisy, right? You want them to all be the same. So you want to control as many things as possible. You want to have the same temperature, okay? So you don't want to put like 2 plants in the refrigerator and 2 plants, in the windowsill, because then you're messing up they're not getting the same amount of sun, but they're also not getting the same amount of the same temperature either. So you want as much as possible the same. That way, if you have as much as possible the same, you can when you're interpreting your data, you can better say, I kept all of these things the same. So whatever caused the difference in, say, the growth of these plants must have been the liquid we used to water them, because nothing else was different between all of these groups. If you have lots of different things between each group, then you don't know, was it, the difference in temperature? Was it the difference in light? Was it a interaction between the liquid and the difference in temperature? So it's really important that you keep as much as possible the same. Now the next thing I'm going to do is I'm going to talk about graphing. This graph, you can see they don't have a title, so you'll really want that. And I'm just going to go quickly because I know we've discussed this before. You want to label the x and y axis. If it needs to have units, like for example this one has years, you want to do that. Otherwise they might think you're talking in minutes or hours or whatever. You can see this graph has the population size. And another thing you can see is, do you see how they're going by 250 every single time? Okay, you want to make sure your numbers are labeled the same. Here they're going by 1 every single time. That's fine. What you don't want to do is have like 1, 2, 5, 6, 10, 11, 15, 20. You don't want your numbers like that. You want them to be, you know, the same like counting scale throughout. For example, 5, 10, 15, 20, 1, 2, 3, 4, 5, 250, 500, 750, 1,000. Okay, you don't just want to put like, some people will, I've seen this before, they'll just take, you know, whatever each data number that comes out, and they'll put the first data number there, and the second data number there, and the third one there, and the fourth one there, and they're not evenly spaced, and then you're not going to be able to see the trends in your data that way. So it's really, really important that you have each axis, the numbers on each axis are evenly spaced. And you can also see not only are they evenly spaced numerically, like 5, 10, 15, 20, but you can see they're evenly spaced space-wise, if that makes sense. So there's like 4 little blocks, and then a number, 4 little blocks, and then a number, 4 little blocks, and then a number. You don't want to have them like all spread out. For example, you don't want to put like your first number here, and then your second number there, and your third number here. You know, that's also going to mess up your data, and it won't give you a good idea of the trend in your data. So that's another thing you don't want to do. The last thing is I kind of want to discuss a best fit line. So when you're making a best fit line, you have your data like this, and this would be a line that's kind of just plotting all your data points, right? It's not a best fit line. One thing I see a lot of people do when they make their best fit line, and I'm going to make this in blue, okay, is they'll just take a line, they'll line it up with their x-axis, and they'll just draw something like that. There's my best fit line. Your best fit line does not always start at the origin, okay? So let me take another color. Let's try this color, whatever. And your best fit line is going to be basically a line that passes as close as possible to all of your data points. So when I'm like really wanting to be particular about my best fit lines, I like to use those rulers that are kind of see-through, because then I can kind of line it up, because you want to use a ruler. You don't want to do it freehand. And you kind of want to have it go. The thing I'm using right now is not a good ruler. It's not a see-through ruler, so you know, your best fit line might look something like that. And it's okay, it doesn't touch the x-axis, that's totally fine. But you want it to be as close as possible to all points. So this is an okay one. It's not excellent because, you know, these 2 points are, you know, really much farther away than all the others. But overall, it's a decent one. It's okay. But that's kind of what your best fit line would look like. It's usually a straight line. Sometimes if you've got logarithmic data, it might look a little differently, but you know, overall it's pretty straight. So here's another one. Again, you know, you don't have to start at 0. You can start kind of wherever. I'm going to try and make this one again. I'm not using a real ruler, so it doesn't look super excellent. I'm not really thrilled with how that turned out, but we'll go with it. Another thing I see people do for best fit lines that is not correct is they make one line that touches the first data point and the last data point. And mine's going to be a little crooked because I said I wasn't using a good ruler. And that's also not a best fit line. A best fit line does not have to touch your first and last points. In fact, if you look at this one, this is a really terrible best fit line, right? Because basically all the data points are on one side of the line. So that's a really bad best fit line. So you wouldn't want to use that kind of best fit line. You know, you don't want to, sometimes it does the first and the last points, but usually it doesn't, and that's totally okay. Sometimes it doesn't even touch any of your data points, and that's also okay. You just want it as close as possible to all of your points. You kind of want to think about this is also kind of silly. You want to think about like your data points are like they have feelings, and you want them all to, you want to feel like you're maybe like treating them all fairly, and you don't want anyone's feelings to get hurt. So you don't want anyone to get like too far away from the best fit line because, you know, it might hurt their feelings. That's also kind of a silly way to think of drawing a best fit line, but it helps me.